Hi, this is Dr. Shanmugapriya, Professor and HOD of Biochemistry at Government Thuthukudi Medical College. I am here to discuss about DNA fingerprinting, principally because there are many occasions where you might have to be able to interpret a DNA fingerprinting report, or you might be asked about this topic. Yeah, so I have few objectives for this session. I am planning to begin the discussion on DNA fingerprinting with an introduction to number 1, restriction enzymes. Yeah, and after you understand restriction enzymes, I'll tell you about hybridization techniques or blotting techniques, because these two are going to be the base of DNA fingerprinting technique when it was first discovered by Jeffrey. Because when Jeffrey thought of DNA fingerprinting, he used southern blotting or RFLP. Yeah, he basically used restriction fragment length polymorphism, which utilizes southern blotting technique for person identification. And after we discuss about RFLP, I'll also tell you about satellite DNA being used for the same purpose. And we call it as VNPR analysis. Yeah, satellite DNA being used for person identification is called as VNPR analysis. So these are the objectives of this session. So I'll be telling about restriction enzymes, and then about southern blotting, and then RFLP, and finally about VNPR analysis. To begin with, about restriction enzymes. So what are restriction enzymes? These are enzymes which cut at specific sites. That is why they are otherwise called as restriction endonucleases. Do you understand this? Exonuclease is an enzyme which cuts an oligonucleotide or a polynucleotide from either the 5' end or from the 3' end. If it cuts from one of the two ends, that's called as an exonuclease. What is an endonuclease? Endonuclease makes only one nick. So you will have to specify the site at which it cuts. So restriction endonucleases are enzymes which cut at specific sites, and those sites are usually palindromic sequences. So what is palindrome in literature? Let me tell you about palindromic sequence later. First I'll tell you about palindrome. So palindrome is a word or a phrase which carries the same meaning when read from either side. Yeah, for example, rotor, R-O-T-O-R, you read it from either side, it's the same, right? Manorama, yeah, again read it from either side, it's going to be the same. So this is palindrome as far as literature is concerned. Now what is the palindromic sequence? Palindromic sequence is a double-stranded sequence wherein as the 3', 5' strand sequence, you have the 5', 3' strand sequence read in the reverse direction. Do you understand this? It's a double-stranded sequence wherein as the 3', 5' strand sequence, you have the 5', 3' strand sequence read in the reverse direction. For example, consider this sequence GG, AT, CC. If this happens to be the 5', 3' sequence, what would be its 3', 5' sequence? Write complementary sequences to all these sequences. For G it is C, C. For A it is T. For T it is A, G, G. Right? So read it from either side, there is no difference. Do you get it? So what is a palindromic sequence? Palindromic sequence is a double-stranded sequence wherein as the 3', 5' strand sequence, you have the 5', 3' strand sequence read in the reverse direction. Now, depending upon how an enzyme cuts this palindromic sequence, restriction endonucleases are of two types. One is a sticky end producing restriction endonuclease, and the other one is a blunt end producing restriction endonuclease. Okay, so what does a sticky end producing restriction endonuclease do? This enzyme cuts both the strands at two different sites. Yeah, for example, if it cuts the top strand after 2 nucleotides, and if it cuts the bottom strand after the 4th nucleotide. Instead of cutting both the strands after the 2nd nucleotide, what does this enzyme do? This enzyme cuts the top strand after the 2nd nucleotide, whereas it cuts the bottom strand after the 4th nucleotide. In this case, the cut ends produced are staggered. Right, the cut ends produced are staggered. So this will be one end and this becomes the other end. In this case, you call it as a sticky end producing restriction endonuclease, because when you bring both these fragments together, they have complementary bases, right? They have hanging edges with complementary bases. So they stick together easily. So if you are trying to form a recombinant vector, yeah, what is a recombinant vector? A vector which has got a gene of interest. Yeah, what is a recombinant vector? Recombinant vector will have a prokaryotic vector that is attached to some gene of interest. In that case, if you use a sticky end producing restriction endonuclease to cut the vector and using the same enzyme to cut the human chromosome, then both the vector and the gene fragment will have hanging edges with complementary bases. So when you bring them close together, they will stick together easily. That is why it is called as a sticky end producing restriction endonuclease. Okay, that is the advantage of using a sticky end producing restriction endonuclease. Whereas what does a blunt end producing restriction endonuclease do? Let's consider the same palindromic sequence, yeah, 3' 5' strand and 5' 3' strand. If the 5' 3' strand sequence happens to be GGATCC, what are the complementary sequences in the 3' 5' strand? CC, TA, GG. Okay, so you find complementary base, you find them to be similar. That's why it's called as a palindromic sequence. Now a blunt end producing restriction endonuclease cuts both the strands at the same point, either after the first nucleotide or after the second nucleotide or after the third nucleotide. For example, if it cuts here, after the third nucleotide, it cuts both the top strand and bottom strand, then the cut ends produced are blunt. Yeah, the cut ends produce a blunt, and this is called as a blunt end producing restriction endonuclease. So which is advantageous? Generally, it's an accepted fact that sticky end producing restriction endonuclease is advantageous because they have hanging edges with complementary bases. So you can link two unrelated genomes to form a recombinant vector. And if you look at Harper, you have been given many examples for sticky end producing restriction endonuclease, and the only one example that is given for blunt end producing restriction endonuclease is HPA1. Okay, that is all about my first objective of me trying to tell you about restriction endonucleases. So what are restriction endonucleases? They cut at specific sites, and those sites are palindromic sequences, and depending upon how they cut the palindromic sequence, they are of two types. One is sticky end producing endonuclease, the other one is blunt end producing endonuclease. Sticky end cuts both the strands at two different sites so that there are hanging edges, they stick together easily. Blunt end producing enzyme cuts both the strands at the same point, so the cut ends are blunt. And one example for blunt end producing restriction endonuclease is HPA1. Now about southern blotting. So southern blotting or southern hybridization. First fact which you should know about this technique is it is a technique that is used to identify unknown DNA fragment. Yeah, southern hybridization or southern blotting is used to identify an unknown DNA fragment. Okay, and it's named after the person who invented this technique. Okay, so what is the first step in southern blotting or southern hybridization? The first step will be to separate all the fragments by electrophoresis. Suppose you have 100 DNA fragments, okay, and you are asked to find out of the 100 DNA fragments which fragment has got the gene of your interest. Your aim is to find out of all the 100 fragments, which fragment has got the gene of my interest. The first step would be to separate all the 100 fragments based on length by electrophoresis. Yeah, I'll tell you what it means. So what I'm drawing here is a gel. To be very specific, it's a polyacrylamide gel which has got 3 wells. Yeah, these are called as wells into which you are going to add the sample. Now you have a sample of 100 DNA fragments. You add all the 100 DNA fragments here. Let me represent the red color fragment. Let me represent the fragment with gene of interest in red color. To find out this fragment, the first step would be to separate all the fragments based on length. That can be done by applying an electrical field. When you connect this gel to an electrical field, you connect it in such a way that the point close to the point of application, the electrode close to the point of application is connected to negatively charged electrode. The opposite side is connected to positively charged electrode. When you switch on the electric current, because all these DNA fragments are negatively charged, they have phosphate groups, multiple phosphate groups. Phosphate groups are negatively charged. So because these fragments are negatively charged, they start moving towards positively charged electrode. The migration rate will be affected by the length of the fragment. Longer fragments cannot move faster, so they stay close to the point of application. Shorter fragments move faster. And let's just take that this third fragment has got the gene of interest. Yeah, we do not know it now. We have just separated all the fragments based on length. So after separation, so what's the first step of sudden blotting or hybridization? It is separation of fragments by electrophoresis. And after separating them, in the next step, you are going to denature all the fragments. So why do you denature all the fragment? Because in the next few steps, you are going to add a probe. Yeah, that probe is going to be an oligonucleotide or a polynucleotide which has got sequences complementary to the gene of interest. And that probe is radio labeled. It is ready to give the signal after binding to the gene of interest. And for that probe to hybridize, these fragments should be single-stranded. Do you understand this? So after separation, the second step is you denature all the fragments so that they are no more double-stranded, they are now single-stranded. After denaturation, you take this gel and you blot it on a nitrocellulose paper. So the third step is called as a blotting step, where you take the gel and you blot it on a nitrocellulose paper. For simple reason, handling gel is not that easy. Yeah, even if you take the gel, you know the gel would break. Okay, so what do? You take the gel and you blot it on a nitrocellulose paper so that all these fragments get transferred to the paper. That's why it's called as a blotting technique. And after blotting, you take the paper. Now it is no more a gel. What you have is a paper. Now you take this paper and you dip this paper in a solution containing radio-labeled probe. Have you seen a Petri dish? You must have worked with a Petri dish, right? So culture plates. So you can take one such Petri dish and you fill this Petri dish with a solution containing radio-labeled probe. And what did I tell you about that probe? I said that probe is labeled with a radioactive substance and it's complementary to the gene of interest. So the fourth step is you dip it in a solution containing radio-labeled probe. Let me show the radio-labeled probe in blue color. And these radio-labeled probes will try to find complementary sequences on all these fragments. Once they find complementary sequences, they hybridize. So the fourth step is called as hybridization step. Why do you call it as a hybridization step? Because in this step, the radio-labeled probe is allowed to hybridize with the unknown DNA fragment. So we know this radio-labeled probe will go and hybridize with the third fragment, third band. So you give enough time for this hybridization to happen. Then you take out the paper and you wash it. Yeah, you take out the paper and you wash it to remove all unbound probes. So what is the fifth step? The fifth step is called as washing step. After washing, you take this paper and you expose it to X-ray. Because it is radio labeled, it forms a marking on the X-ray. So this step is called as autoradiogram. Yeah, this step is called as autoradiogram. By visualizing the X-ray, you will know which fragment has got the gene of interest. And this is called as Southern blotting or Southern hybridization. So what are the facts you need to know about Southern blotting or Southern hybridization? This technique is used to identify unknown DNA fragment. Among all fragments, you are trying to identify one specific fragment that is done by Southern blotting or Southern hybridization. Now, what are the steps involved in this? The first step is you separate all the fragments based on length by electrophoresis. Second step, you denature all the fragments. Only then these fragments can hybridize with the probe. After denaturation, you take the gel and you blot it on a nitrocellulose paper. So the third step is called as a blotting step. After blotting, you dip the paper in a solution containing radio-labeled probe, during which time these radio-labeled probes have sequences. They try to hybridize with the bands if they have complementary sequences. After they find the fragment with complementary sequences, they hybridize. So the fourth step is called as a hybridization step. And after you give enough time for hybridization, you take out the paper, you wash away excess unbound probe, and you expose the paper to X-ray. That's called as autoradiogram. By visualizing the autoradiogram, you will know which fragment has got the gene of interest. This is Southern blotting or Southern hybridization. If you are using the same technique, same principle, but for identifying unknown RNA fragment, that is called as northern blot. Okay, if you are using the same technique and the same principle, but for identifying unknown RNA fragment, that's called as northern blot. So what do you think will be the difference between southern blot and northern blot stepwise? I said southern is for DNA identification, northern is for RNA identification, but stepwise, what do you think will be the difference? Do you think northern blot will involve a denaturation step? Because RNAs are already single-stranded, right? In that case, you don't have to employ a strong denaturation step. Still, in northern blot, there will be a milder denaturation step. And the purpose of having this milder denaturation step is to remove hairpin loops. Anything which is single-stranded would have got hybridized within itself to form a hairpin loop. And until there is hairpin loop, it cannot hybridize with the probe. So there should be a denaturation step even in northern blot, but it will be a milder denaturation step. Okay, that's the difference between southern blot and northern blot. And if you're using the same principle, but for identifying an unknown protein, that is called as western blot. Okay, if it's used for identifying unknown protein, that's called as western blot. So what do you think is the difference between southern and northern blot on one hand and western blot on the other hand? I said western blot is for protein identification, whereas southern and northern blot would be for identifying oligonucleotides or polynucleotides. So be it southern or northern blot, the probe will be a cDNA, complementary DNA, which is complementary to either DNA or RNA of interest to you. So what is the probe that is used in southern and northern blot? It's a complementary DNA. Whereas if it's western blot, for you to identify an unknown protein, can you use a cDNA as a probe? You can't. cDNA is not going to go and bind to the protein. So in western blot, to identify an unknown protein, you synthesize antibodies. You synthesize monoclonal or polyclonal antibodies which are directed against the protein of interest to you. You label them and use them as a probe. So what is the probe that is used in western blot? It's an antibody probe. Okay, that's all about southern blotting, southern hybridization, and then northern blot or northern hybridization, western blot or western hybridization. So we have learnt about restriction enzymes, we have learnt about southern blotting and southern hybridization. Now about the topic proper, it is DNA fingerprinting. So what is dactylography? You must have learnt about this, right? What is dactylography? It is person identification. As the name indicates, it is person identification by utilizing uniqueness of fingerprints. That we know, right? What is dactylography? It's person identification by utilizing the uniqueness of fingerprints. So what is DNA fingerprinting? DNA fingerprinting is again person identification by utilizing some of the unique sequences of DNA. Do you understand this? So what is DNA fingerprinting? It is person identification by utilizing some of the unique DNA sequences. And as I told you when I started the session, it was first identified by Jeffrey. And when Jeffrey identified it, he used RFLP or restriction fragment length polymorphism for person identification. Okay, so what was used for person identification to start with? Even now in many centers they use RFLP or restriction fragment length polymorphism for person identification. So what is the principle behind RFLP being used for person identification? The principle is, listen to this, the principle is for no two persons, number and size of restriction digest. Yeah, for no two persons, number and size of fragments in a restriction digest can be identical. It's very simple. For example, if I am using a restriction endonuclease which cuts at GATC sequence. Okay, I am using a restriction endonuclease which cuts at GATC sequence. In my genome, the first GATC might be present as 100th to 104th nucleotide. The second GATC might be from 200 to 204. Third might be from 300 to 304. It will not be equally spaced like this. I am saying this just for you to understand. Okay. Whereas for another person, person B, the first GATC might be from 500 to 504. Second GATC might be from 1000 to 1004. Third might be from 1500 to 1504. So what do you think about my restriction digest and what do you think about the other person's restriction digest? In my genome, GATC is repeated frequently. So my restriction digest will be short. The fragments will be shorter and there will be more number of fragments. Do you understand this? I'll have shorter fragments and more number of fragments. Whereas person B, the fragments will be longer and the number of fragments will be less. So because location of restriction site, because location of restriction site in two genomes are not identical, number and size of fragments in a restriction digest of no two persons can be identical. Do you understand this? So this is the basis of RFLP being used for person identification. For example, from the crime site, from the crime site they have obtained a blood stain. Okay, they have obtained a blood stain. And there are two suspects, person A and person B are the two suspects. And you are asked to find out who could have committed the crime. Okay, what would be the first step? The first step would be to collect blood sample from person A and person B. And from both these blood samples, you extract DNA. So now you have 3 DNA. How do you have 3 DNA? 2 from 2 suspects and 1 from the blood stain obtained from the crime site. Okay, let me show person B's DNA in a different color. So now you have 3 DNA samples. So what is step number 1? You collect blood sample from the 2 suspects. From the 2 blood samples and from the blood stain, you extract DNA. So now you have 3 DNA samples and you are subjecting these 3 DNA samples to digestion. All these 3 DNA samples are subjected to digestion by the same restriction endonuclease. You are not changing it by the using the same restriction endonuclease, but the cut fragments will be different because location of restriction sites will be different. So after restriction digestion using a restriction enzyme, you separate all these fragments based on length by electrophoresis. So what I am drawing here is again a polyacrylamide gel with 3 wells. Yeah, let me name these wells as A and then blood stain and then suspect B. So into A well, I have added restriction digest of person A's blood sample, and into the blood stain I have added the restriction digest obtained from the blood stain, and finally person C's restriction digest. Now I am applying an electric current. Close to the point of application is connected to which electrode? Negatively charged electrode. The opposite side is connected to positively charged electrode. Then I am switching on the electric current because all these DNA fragments are negatively charged. They start moving. Longer fragments stay close to the point of application and shorter fragments move faster. Okay, similarly blood stain restriction digest also would have got separated, and person B's. So now you know the conclusion. What is the conclusion? You see that person B's restriction digest matches that of the blood stain. Yeah, which means person B has come to the crime site. You cannot say you have committed the crime. All that you can say is you have come to the crime site, that's why your blood sample is found there. Okay, so this is how RFLP. Why am I saying it is RFLP? Because I used a restriction enzyme to cut all 3 genomes, and I've tried comparing the restriction digest fragments, length and size across these 3 samples. Yeah, that's why it is called as RFLP. And this is used, or this was the only technique that was initially used for DNA fingerprinting when Jeffrey identified it. Later on, we found out something called as satellite DNA. So now I am going to tell you about something called as VNPR analysis. Yeah, I am going to now tell you about something called as VNPR analysis. And VNPR analysis is nothing but variable number of tandem repeats. Yeah, so this is variable number of tandem repeats analysis, which is based on satellite DNA. So what are the facts you know about satellite DNA? I'll tell you the basis first and then I'll tell you how it is being used for person identification. So satellite DNA is one of the miscellaneous sequences. Yeah, it is one of the miscellaneous genomic sequences, and they are otherwise called as highly repetitive sequences. Please memorize these names. It is otherwise called as highly repetitive sequences. They are also called as simple segment repeats. Yeah, they are also called as simple segment repeats. So they are one of the types of miscellaneous sequences found in human genome, and they are otherwise called as highly repetitive sequences or simple segment repeats. So why are they called as simple segment repeats? Just to tell you that they are simple segments which are just repeated, they carry no meaning. Yeah, which means they are totally non-coding sequences. That's why I said it comes under miscellaneous sequences. Okay, and why are they called as highly repetitive sequences? Because if you take the whole human genome and if you count the number of times they are repeated in one genome, in one human genome, if you count the number of times they are repeated, it is repeated more than 10 power 6 times. Because it's repeated more than 10 power 6 times, you call it as a highly repetitive sequence. And when you know they're completely non-coding, why are you learning about them? It is because though I said they are repeated more than 10 power 6 times, not all the 10 power 6 repeats will be concentrated at a single locus. They are distributed at various loci. Yeah, for example, if you take CAG repeat alone, the CAG repeat is repeated more than 10 power 6 times in your genome and my genome and everybody's genome. But not all the 10 power 6 repeats will be in one locus. It will be repeated, it will be distributed at various loci. And at every locus, the number of times it's repeated is found to be unique to a particular person. Yeah, for example, if you look at the CAG repeat at one locus, you can have any locus, some number. Yeah, at that locus, if you count the number of times it's repeated in your genome, it can be 8, it can be 9, whereas in mine it's going to be 7 or 10. So it is not common to all, it is found to be unique to a particular person. And that is the basis of satellite DNA being used for person identification. So what is the basis? Though they are repeated more than 10 power 6 times in the whole human genome, the number of times they are repeated in a particular locus is unique to a particular person. And this has been used to clear off many paternal dispute cases. Yeah, it has been used to clear off many paternal dispute cases. I'll give you an example to tell you how it works. Listen to this. None of the textbooks explain you how it is being used for person identification. They just tell you satellite DNA is used for person identification. And not knowing how it is used, it is not practically possible for any human being to just have it in memory. So listen to this. This is how it is used to clear off paternal dispute cases. Suppose there is a mother. Yeah, suppose there is a mother with a child, and this mother claims person X as the father of this child, which obviously person X does not agree. The person X denies it, and you are asked to find out if X can be the biological father of this child. Do you understand the scenario? There is a mother with a child, and this mother claims person X is the father of the child. X denies to this claims, and you are asked to find out if X can be the biological father of this child. What will be the first step? The first step will always be to get a sample from all the three of them. So I'm going to get a blood sample. So first step is I get blood sample from the mother, from the child and the suspected father. From all three samples, I've extracted DNA. Yeah, from all three samples, I've extracted DNA. Now, in all these three DNA samples, I'm going to check at least 5 microsatellite repeats at 5 loci each. Yeah, this is how all forensic laboratories work, because just by looking at one repeat at one locus, for example, as I said, CAG repeat at one locus, just because in your genome it is repeated 7 times and in my genome also it happens to repeat 7 times, you cannot say you and me are the same. Do you understand this? You cannot you cannot get yourself to a conclusion just by looking at one repeat at one locus. There is a very high probability that many people have the same repeat, same number of repeat for a microsatellite repeat at a particular locus. So what do? We look at least 5 microsatellite repeats at 5 loci each. So totally you look at 25 sites. Okay. For example, CAG repeat at one locus, say A locus, yeah, CAG repeat at one locus, you are checking the child's DNA and you are saying how many times it's repeated. Yeah, you are checking the child's DNA, you are saying how many times it's repeated. Suppose child's DNA shows 8/8 repeats. Okay, child's DNA shows 8/8 repeats. What does it mean? I said only one repeat and I said I'm looking at only one locus, but I'm giving you 2 numbers, 8/8. What does it mean? Why do I have 2 numbers? It is because you have 2 copies of every chromosome, don't you? Chromosome 1 you have 2 copies, chromosome 2 you have 2 copies, chromosome 3 you have 2 copies. So when I say a locus, that locus will be present in both the chromosomes 1, be present in both the chromosomes 2. Do you understand this? So every locus there are 2 places. So 8 bar 8 repeats means in one chromosome it's repeated 8 times, in the other chromosome also it is repeated 8 times. Now for the same repeat, same CAG repeat at the same locus, you are checking in the maternal DNA. Okay, suppose maternal DNA shows 7 bar 8 repeats. Maternal DNA for CAG repeat at one locus shows 7 bar 8 repeats. So what is your inference? Your inference is one of the 8s of the child. Yeah, one of the 8s of the child has come from the mother. Yeah, it means one of the 8s of the child has come from the mother. So it's imperative that the other 8 of the child comes from the father. Okay, suppose paternal DNA shows 7 bar 9 repeats, or it shows 10 bar 12 repeats. What will be a conclusion? This person cannot be the biological father of this child because his repeat number does not match with what is expected based on the child's DNA repeat. Okay, so this is how satellite DNA is being used for person identification. So I am repeating it. Satellite DNA is otherwise called as highly repetitive sequences. It is also called as simple segment repeats because they are totally non-coding. Why do you call it as highly repetitive sequences? Because it's repeated more than 10 power 6 times. Okay, and why are we using satellite DNA for person identification? Though we say that it's repeated more than 10 power 6 times, not all 10 power 6 repeats are concentrated at a single locus. It is distributed at various loci. Okay, and at every locus, the number of times it's repeated is found to be unique to a particular person. Yeah, that is the basis of satellite DNA being used for person identification. Okay, and it's being used to clear off paternal dispute cases. For example, there is a mother with a child and mother claims person X is the father of the child, X denies. You are asked to find out if X can be the biological father of the child. In that scenario, what would you do? You get blood sample from all three, from the child, from the mother and the suspected father. From all 3 DNA samples, you extract DNA, and all 3 DNA samples, you look for at least 5 repeats at 5 loci each, which means totally you look at 25 loci. Okay, for example, 1 repeat at 1 locus, the child's DNA shows 8 bar 8 repeats, which means one chromosome shows 8 repeats, the other chromosome also shows 8 repeats. Now the same repeat, same locus, look at the maternal DNA. Suppose maternal DNA shows 7 bar 8 repeats. It means one of the 8s of the child has come from the mother. So the other 8 of the child should come from the father. You are looking at the father's DNA. As I told you previously as an example, if father's DNA shows something other than 8, 7 bar 9 or 10 bar 12 or 15 bar 16, what will be a conclusion? No, this person cannot be the biological father of this child. Suppose father's DNA shows 8 bar 6 or 8 bar 10 or 8 bar 12. You will say, yes, this person, there is a possibility that this person is the biological father of this child. Yeah, when you look at 25 sites, you will have a clear-cut conclusion. This is only by looking at one site. But what do? We look at least 25 sites. So by the end, we'll have a conclusion or a concluding evidence. Okay. And about satellite DNA, they are of 2 types. Okay, satellite DNA is of 2 types. One is microsatellite repeat and the other one is mini satellite repeat. Based on length, there are 2 types of satellite DNA. One is microsatellite repeat, the other one is mini satellite repeat. And the other name for mini satellite repeat is variable number of tandem repeats. Yeah, the other name for mini satellite repeat is variable number of tandem repeat. That is why it's called as VNPR analysis. Just because I said satellite DNA is used for person identification, you should not fumble if somebody says somebody asks you what is the purpose of VNPR analysis. You should know both are same. Satellite DNA, one of the types is mini satellite. It's otherwise called as VNTR or variable number of tandem repeats. So what I've told you so far is related to DNA fingerprinting. And for you to understand DNA fingerprinting, I started with restriction enzyme. I said it cuts at palindromic sequences. I said there are two types, sticky end producing enzyme and blunt end producing enzyme. And then I told you about southern blotting or southern hybridization, because that is the basis of Jeffrey's technique of DNA fingerprinting. So southern blotting or southern hybridization will be used for identifying unknown DNA fragment among all fragments. Okay, and for this the first step is separating all the fragments based on length by electrophoresis. You denature all the fragments. You blot the gel on a nitrocellulose paper. That's why it's called as a blotting technique. You dip the nitrocellulose paper in a solution containing radio-labeled probe, and you allow the radio-labeled probe to hybridize with the specific fragment. So the fourth step is called as hybridization step. After you give enough time for hybridization, you wash away excess unbound probe and you expose it to X-ray. That's called as autoradiogram. By visualizing autoradiogram, you will know which fragment has got the gene of interest. Northern blotting is used for identifying unknown RNA, and because RNAs are mostly single-stranded, the hybridization step, the denaturation step in northern blotting will be milder. Yeah, again, why do you employ a denaturation step at all? Because single-stranded RNAs would have formed hairpin loops. With hairpin loops, they won't be able to hybridize with probes. Okay. If the same technique is used for identifying unknown protein, that's called as western blot. And the difference between western blot on one hand and southern and northern blot on the other hand is the probe that is used. In southern and northern blot, the probe that is used is a cDNA, which is complementary to either DNA or RNA of interest. Whereas in western blot, the probe that is used is an antibody. Okay, so that's all about blotting and hybridization technique. And then I told you about DNA fingerprinting, which is nothing but person identification by utilizing uniqueness of DNA sequences. And this was first identified by Jeffrey, and he used southern blotting or southern hybridization. And the basis of southern blotting being used for person identification is that for no two persons, number and size of fragments in a restriction digest can be identical. Yeah, this is called as RFLP or restriction fragment length polymorphism. And these days we have started using satellite DNA, and the other name for mini satellite is variable number of tandem repeat. So VNTR analysis is also used for person identification. Okay. So if you know these facts, that would be more than enough. And let's discuss few MCQs to understand how much you have understood. Restriction endonuclease function true or all except. Does the enzyme cut both the strands of double-stranded DNA? I showed you clearly that it cuts both the strands. The cut ends produced are sticky, sometimes yes. The cut ends produced are blunt, sometimes yes. Do these enzymes cut single strands of DNA? Do they cut single strands of DNA? No. So true or all except, your answer should be it cuts single strand of DNA. Correct statement regarding restriction endonucleases. Does it cut at short sequences of DNA? Yes. Short specific palindromic sequence, yes. Does it act at 5', 3' direction, or does it act at 3', 5' direction, or does it act in both the directions? I am giving you 30 seconds to think and answer. Yeah, think and try to answer to yourself which of these would be right. It's a PGI question. Does it act at 5 prime, 3 prime, or 3 prime, 5 prime, or in both the directions? None of these, right? Directions can be specified only for an exonuclease. Only if it's an exonuclease, you will have to specify whether it cuts from 5 prime end or it cuts from 3 prime end. But here it's an endonuclease, which makes only one nick. So for endonucleases, there can be no directions. For an endonuclease, it's only the site at which it cuts. Okay. Probe used in Western blot is, I told you repeatedly, it's antibody. DNA fingerprinting is identified by Jeffrey. True statements of DNA fingerprinting are all except. So what are the two techniques used for DNA fingerprinting? I said one is restriction fragment length polymorphism, the other one is satellite DNA. Okay, and restriction fragment length polymorphism employs southern blot. It employs southern blotting technique. So true or all except. Does it involve southern blot? Yes. Does it involve northern blot? What is northern blot? I said it's identifying RNA fragments. You cannot differentiate a person based on RNA sequences because most of these RNAs are spliced. So most of these RNAs would have lost their variable sequences. So northern blot is never a form of DNA fingerprinting technique. Does it involve gel electrophoresis? Yes. Does it involve RFLP? Yes. DNA or RNA fragment which is radioactive or chemically reactive to detect a specific fragment is. Obviously it is a probe. Thank you.